Welcome, thanks for joining us. We'll get started in about 5 minutes. Hi, everybody. We'll get started in about 3 minutes. Hi, everybody. It's just about 3 o'clock. Thank you for joining us for our Sci Cafe series. Today's talk will be recorded and available on apalachicola.reserve.com. If you have a question, please raise your hand, or you can put it in the question function, and we'll ask our speaker. Our speaker today is Jill Fleiger, and she is the environmental administrator with the Florida Department of Agriculture, Division of Aquaculture. And she's going to talk to us today about water quality and water testing in Apalachicola Bay. We will have time at the end for questions, but if you want to put your questions in the question area, we'll be happy to ask them at the end. So take it away, Jill.
Thank you. Now I got to figure out how to share my screen again. I'm not seeing. Oh, here we go. I think it's coming. Then.
Yeah, you're just about, there you go.
Oh, I changed the screen. Give me 1 second. Okay. So that's good. Everybody can see the first slide.
I can see it.
Looks great, Jill.
Awesome. Well, thank you for inviting me to kind of go over the Shellfish Harvest Area Classification Program. I try to keep it pretty specific to the Apalachicola region. But I did want to start out, I like this as a title slide because it really highlights how much area this program covers in the state of Florida. And we have a really specific purpose, with water quality monitoring and how it relates to shellfish harvesting. And so from this map, you can see we cover pretty much all the way over from Pensacola, down around the Panhandle, Big Bend, down to 10,000 Islands in Southwest Florida. And then we kind of skip over the Keys. There's not a lot of oyster and clam resource down there historically. And then we pick it back up in St. Lucie County, all the way up to sort of the northern part of Jacksonville. And we have 5 field offices, 1 in Panama City, 1 in Apalachicola, 1 in Cedar Key, 1 in Port Charlotte, and 1 in Melbourne. We have 10 staff that are responsible for collecting and managing the harvest areas throughout the state. So we cover a lot of area, and we have about 1.1 million acres or so classified within the state. So I just wanted to give a little background for the first few slides about why molluscan shellfish are so regulated, how it's kind of structured, and why we do what we do. So molluscan shellfish are known to be filter feeders. And so from that, they can retain microorganisms, that, including toxins and chemical contaminants that they take up in their surrounding. And so if the water is contaminated, this could be a potential health hazard to consumers. In a very common way, or most popular way to eat shellfish among the consumers is raw or partially cooked. And this industry is very important economically, so we want to be able to outweigh some of these safety risks with consumption, but also be promoting the industry as a whole. So the way we do this is we are part of the National Shellfish Sanitation Program, and this makes up different state authorities, federal partners, including the EPA and the FDA. Industry partakes in this as well, and also some academic institutions. And so what happens is we all get together in what is called the Interstate Shellfish Sanitation Conference. And we establish minimum requirements to regulate the interstate commerce of shellfish. So we come up with a document called the NSSP Model Ordinance. And so this is to ensure that we protect public safety by ensuring that the harvest areas are properly classified, and also ensure that shellfish are handled properly at all levels, from harvest all the way to the final sale to the consumer. And the US FDA is it's a partnership, but they also oversee and ensure that states are complying with the NSSP requirements. And they do this by annual audits. So in the state of Florida, the shellfish that are most commonly regulated are oysters, clams, and I did include mussels, but I put an asterisk there because there isn't a huge mussel, like recreationally harvested mussels, and there's really no commercial. So it's mainly just oysters and clams. Before I get into sort of the whole process of how we do our classification and our management, I wanted to go over some of our goals of what we are looking for when we're classifying and managing our harvest areas in the state. So we want to maximize the acreage to allow for harvest, and we also want to minimize the number of days they're going to be closed, while we're also protecting the health of consumers from shellfish-borne illnesses. So there's a lot of sort of symmetry that you have to go through to make sure that we're giving the maximize amount acreage, but also making sure that we're protecting the health of the consumers. So the way we do this is called a comprehensive shellfish harvesting area survey. And there's three main components to it. One is what we call the pollution source survey. And I'll get into all of these in the next few slides. We also have a water sampling program, and we also use statistical analysis to determine our classifications in our management plans. So I like to start out with the pollution source survey. This is kind of the first step. In areas that already have this established, it's kind of just a verification. But these are kind of the steps we would take if somebody wanted us to classify a new area, if there's enough interest for that water body. So what we do is we identify all potential direct and indirect pollution sources within a drainage basin surrounding the harvest area. So we kind of pick a survey area around sort of the watershed, and then we go and identify all of the potential pollution sources and map them out. And some of these pollution sources can include marinas, wastewater treatment plants, plant facility outfalls, septic tanks, runoff, and wildlife. And we used to do this as a visual verification where we would go out and do this visually and kind of document all of the different potential pollution sources. Nowadays, we're able to kind of use Google Earth to verify some of those. And we also try to work with state and local agencies because a lot of people kind of have this already in their ArcGIS databases. So we're always looking for new and improved ways to kind of help us do our pollution source surveys. And from there, so just an example of one of our pollution sources, this is actually Lemon Bay. I like this map for this purpose because it kind of shows you how many marinas we can have to document. So if we have a marina or a wastewater treatment plant with a surface water discharge, we have to create these buffer zones around them. And we have a statistical program where we put in like a fecal load, how the water depth, and different things, and then we'll create these buffer zones. So pretty much what that means is anything that's in that buffer zone is going to have to automatically be prohibited. So we won't allow for shellfish harvesting. So this really kind of helps us begin to look at our classification map. So all those red circles, filled in circles, are sort of what is called a marina buffer zone. And so all of those, it will always be prohibited, even if we did take water quality and, if we were 150 feet off. This is a requirement by the National Shellfish Sanitation Program to create these buffer zones. So that is actually, it looks simple, but that can take some time for us to get through. And so while we're doing that, we're also conducting routine water sampling in the shellfish harvesting areas. And these data sample stations are set up outside of pollution sources, also around wild resource, different oyster reefs. And now since aquaculture is kind of coming in, we are also trying to establish some water sample stations around them as well. And then we also do water quality data management, and that helps us open and close the harvest areas. In all of our water samples, we have a dedicated lab in Apalachicola. They run all the water samples for the whole state. The Apalachicola office is lucky enough that they can just drop them right off at the back door, and everybody else actually has to ship to them. So before I get into sort of our water quality, I want to just take a step back and talk about our indicator species. So what we are looking for is fecal coliform, and this is what we test for. So it's a group of bacteria that is excreted in the feces of warm-blooded animals, including humans, domestic animals, and wildlife. And we use it as a way that if it is present and it's at levels that could be harmful to humans, that we also assume that human pathogens could also be present. So this is a map. I just wanted to kind of take it back to Appalach. I relate this map because it shows our classifications and sort of boundary lines, and then all of our water sampling stations. So these are all the water sampling stations within the Apalachicola region. They have quite a few stations. And the section that's in the middle that has water quality stations, but it doesn't have any color coding, those would be the summer areas. But since there's been that 5-year ban, we just haven't really switched over because there's no leases in there and there's no wild harvesting. And so we've been kind of just showing and displaying this winter map. And I'll get into the classifications and the color schemes and sort of what that means. So, but you can see there's probably, I think off the top of my head, there's well over 80 stations within the Apalachicola region. So our water sample collection, we just use whirl packs, 100 ml whirl pack. So it's pretty simple and easy. And we take it in the first 2 feet of the water column. So we just scoop it in, and then we make sure there's headspace in it. We whirl pack it up, and then it's put on ice right away. We actually did these pictures with our marketing group. I thought it was really cool that you get an underwater photo that actually showed us taking the sample. It was pretty neat. So a little bit about our lab. So we have a dedicated lab with a lab manager and 3 lab technicians located in Appalach. All water samples are shipped overnight unless you're actually in the Apalachicola office. And the samples must be maintained between 0 and 10 degrees Celsius. Sometimes in the winter, they'll freeze on us. Sometimes in the summer, they're over the 10 degrees and we can't analyze them. And we use what is called the MTEC method. It's just a direct plating method. So we filter them after collection. We send them off. The technicians filter them. You can see the middle picture. One of our lab techs is filtering it into a glass beaker, and then it passes through the filter. And then from there, they put them into an incubator for 24 hours, and then they count the number of colonies. And then all results are emailed back to the regional offices to figure out data analysis, if they can reopen areas that have closed already. So we have a pretty good operation down there. I think they can do about 150 samples a day, 150 to 175. So once we all collect the water samples and we get the results back, what do we actually do with this data? And so data taken at each sample station is then entered into a database. And we also take salinity, temperature, dissolved oxygen, and pH. And we take salinity, temperature, and dissolved oxygen at the surface and at depth. We also record daily rainfall and river levels. And these are at specific gauges throughout, I guess, the survey area. I think our river gauge is up near Blintstown. And then they are also entered into a database. And then the analysis allows us to look at water quality trends over time and spatially, which is nice. And then it helps us determine the appropriate classification of the harvest area. So I'll go in a little bit about what the analysis is. So we use SAS to determine our classifications. Right now I'm trying to switch us over from SAS to R. So this is a huge project that we're right in the middle of. And so what that does is it brings in our rainfall and our river data and our fecal coliform and temperature and pH and salinity, and it merges them in to sort of one data set that SAS is looking at. And it can look at correlations between rainfall and river levels and bacteria levels at all of the stations. And so from there, we determine the most significant model station. And so, if one zone has 12 stations, we pick the most significant model station within that, and that sets our management plan. And this is what was going to close harvest areas. It's kind of a way we predict that fecal coliform levels could be elevated after a rainfall or after river levels start rising. And then it also allows us to look at trends over time. So for example, is water quality improving or is it degrading? Within the harvest area, we can look at individual stations to see if maybe over time they're degrading and we might have to like reclassify, but we can also sometimes look at them and they're improving and then maybe we can open up more area. That's usually what we're going for. We never like seeing it degrading. And this also helps us set our harvest area classifications. And so we have 5 different classifications, and they kind of determine the harvesting restrictions that are put out there, whether you can harvest or if you have to deprecate. And I'm going to go through each of those on each other slides right after this. And so it's really important to understand that our classifications are actually based off of fecal coliform water quality and also proximity to the pollution sources. So our first and most covenant classification is approved. And this just means that this is the Apalachicola region. I put arrows to show they're always blue. We have the same color coding through the whole state. And so they're usually not near pollution sources. So in Apalachicola, you can see 1601 and 1611. I know you can't see the labels, but the 2 blue areas are the furthest away from the river. And the river is really one of the major inputs into the system. And so usually, generally, these areas are not near pollution sources. It also means that they meet the NSSP water quality standard for fecal coliform at all times. Under adverse pollution conditions, the only time they ever close are during emergencies. And so that could be for harmful algal blooms. Hurricanes and flooding are usually the most common. Storm surge problems, or if we do have a hurricane or tropical storm come through. And this is the most desirable classification type, but it's really hard to attain. And we have about 20% of our total acreage is classified as approved. And then the next one, and they're kind of like in steps of water quality, like not as good water quality. And so the next one, and this is the most common, is conditionally approved. It meets the NSSP water quality standards for fecal coliform when the harvest area is in open status. And so our management plans that we built are used to predictably close when environmental variables such as rain or river increases fecal coliform in the water or in that particular zone. And then we sample, we can only reopen these after we sample and the results show that the fecal coliform levels are at acceptable levels. The most common classification type is about 55% of our total acreage classified statewide.
And Jill, is that the green?
Yeah, that's the green, sorry. I think I tried to make it the right-hand side the same color, but it was like locked in, but yes, that's the green areas. In each green area in this map, you can kind of see lines going through. We call those boundary lines. So each of those zones will have a different rainfall management. And so normally, I know this area quite well. So the one that's closest to the river, if I had a little pointer, I don't know if you can you see my pointer on the screen?
Yes, yes, we can see your person.
Okay, so the one that's closest to the river is probably going to have a rainfall management plan with less rain. So it may close on like 2 inches of rain. And then this zone that's further away may close on like 4 inches. I'm not giving you the right actual rainfall management plan. I can't remember all the zones of rainfall management off the top of my head. And then 1642, which was a pretty popular spot in Appalach before the 5-year ban. it probably had an even better management plan. And so in Apalachicola, there's kind of a north-south and east-west influence from that river. And you kind of get better management as you move away from it. And then we go into, I kind of lump restricted and conditionally restricted together. We don't have a lot of acreage, a lot of areas that are restricted or conditionally restricted. So these are the orange section. So this means that it does not meet approved or conditionally approved water quality standards. So if we did have it open, historically, these were used for relaying. If anybody's not familiar with that, what happens, they would take shellfish from the conditionally restricted, move it into conditionally approved, let it sit for 21 days, and then they could go back and harvest it. That hasn't been a common practice since I've been the administrator. There's been a ton of stress on the resource. People have tried to keep it so that they could see the rest of the areas, and we just don't really have any deprivation facilities right now. I think we have a couple people looking at, starting up deprivation facilities, but they just don't exist at the time. There's like a lot of different things you have to do to be a deprivation facility on your water and testing, and my counterpart program, the processing facility, usually works with them. And then this is only about 5% of our total acreage classified statewide. And then the red zones, which are prohibited, are closed all the time because they're either too close to a pollution source or the water quality just isn't good enough to make it restricted or approved or conditionally approved. We do look at trying to upgrade those areas sometimes. Sometimes if they're closed or they're prohibited because there's a surface water discharge from a wastewater treatment plant, and then they shut that down and go to land-based. Sometimes we can open up these prohibited areas. In Apalachicola, sort of that north section by the river, that's all pretty much dominated by the river flow. And so that will always be prohibited. But yeah, if we can upgrade them, we try our hardest to do that. And that's about another 20% of our total acreage classified statewide. And then we have unclassified areas, and a lot of people get like confused about this. I get a lot of calls. So these are closed to harvesting at all times. These are just areas that we don't have historical data for, or we just don't have a program set up. And I kind of zoomed out a little and tried to show the kind of the section in between Apalachicola and Alligator Harbor. So that isn't classified at all, and people shouldn't be harvesting out of those. So some of the management types of our harvest area. So we have our classification built. We have our zones. You can see Apalachicola was kind of cut up into different classification zones. And so that kind of tells the harvesters where they can harvest. And so now I'm going to get into the when can they harvest. So we have these management plans that are based off of rainfall or river levels. So when those exceed, we have to close the harvest area, and we have to give FWC and the stakeholders, 24-hour in advance. We try our hardest. We try to get our rainfall and river level data in the morning and let them know that it'll be closing at sunset. Sometimes we just get them in late, or the rainfall hasn't been updated to the afternoon. But once it exceeds, we close it. We let FWC know, and all the oystermen, leaseholders. And then, they can kind of plan accordingly. And then we also have emergency conditions. So in the last 5 years, we've had to do a few emergency closures with all the tropical storms and hurricanes. If it's not coming up in the Panhandle, it usually happens on the Atlantic coast. So I'm always looking at tropical storms and also sewage spills. They get some areas have sewage spills or wastewater treatment failures more than others. That will also close the harvest area down. And then, of course, harmful algal blooms. This is a big program that we're trying to improve and watching pretty closely now. And so the three major algal species that we are sampling for, and we work with FWC down in St. Pete. They're the ones that actually do our cell counts and our meat toxin analysis, is Carenia brevis. So that's known to cause neurotoxic shellfish poisoning. I think you guys just went through a red tide bloom back in December. I think the whole Gulf state went through a red tide bloom. It was pretty big. So, and then we look at pseudonitsia, which is a diatom, and that can cause amnesic shellfish poisoning. We're kind of lucky in the state of Florida. Most of the time, the species that we see are non-toxin producing. I could probably do a whole other science cafe on the different kind of harmful algal blooms. And then we also look at pyridinium helminths, and that seems to be more on the Atlantic coast, and that can potentially cause paralytic shellfish poisoning. So once we close them for one of those, 3 or 4 reasons, what do we do? How do we get them reopened? And so we try our best to get out there as soon as we can, based off of weather conditions, time of year. We know that the winter usually doesn't have passing water as quickly as in the summertime. And so we'll go out and we'll sample all the stations within that zone. And then once they show that they have passing results and the environmental conditions are improving, we can reopen them. And then for some of our emergency conditions, such as storm surge, if a major hurricane comes through, if we have a sewage spill in the area that closes it, we will collect shellfish meat samples as well, and they also have to pass. Those aren't as common, that they only happen probably, in Apalachicola, it doesn't happen very often that we have to get shellfish meat to pass, but if we have a storm surge problem, we do. And then for harmful algal blooms, we actually test for toxin in the shellfish meat, and they also have to pass toxin testing in order to reopen. So we have all that set. We have our classifications, we have our management plans. So how do we determine if this is all working? So the National Shellfish Sanitation Program has 2 sampling strategies that states can choose from, and Florida does what is called the Adverse Pollution Condition Sampling Strategy. And so we're required to sample each harvest area zone at least 5 times per year and we set the APC conditions when we're doing our statistical analysis. And this has to be when it's in the open status. So it's the APC condition, the way I like to explain it is it's not quite, it can't be over the closure level, but it can't be 0 inches of rain. And so we try to it's, in between 0.1 inches and whatever the management plan is. And we consider that adverse pollution conditions. So that usually occurs after a rainfall event, or if the river levels are elevated, but not elevated enough to close it. And this helps us determine if the area is in compliance with the water quality standards under the current management plan. We also update all of our data analysis yearly in our annual reports. And then we reevaluate the pollution sources every 3 years to make sure there's no changes. Like Marina's being added in, wastewater treatment plants ask for, higher volume, like things like that is what we're looking for. And we actually work with wastewater treatment plants to get their, or actually the DEP to get their annual reports so we can look at them. And then this comprehensive shellfish harvest area survey, we redo that every 12 years. And so I've moved it to every 9 because we're doing a triennial, kind of a pollution source update every 3. I don't like getting put, in the corner of like, okay, it's 12 right now, because if you miss that mark, FDA will say you're out of compliance. So I've kind of been pushing us more to every 9 years. If an office is going through something, like what our office in Panama City went through Hurricane Michael, and so I didn't push them too hard on the 9 year because we do have till 12 years, and so sometimes that can shift a little bit based off of what's going on at each office. But we try to at least hit that 12-year mark. So we stay in compliance with the program. And that the 12-year surveys allows us to move boundary lines, change zones. If things have improved, we can open up more area. That's kind of the time to do that. Change the management plan if we can get a better management plan. We usually try not to make it worse. We try to make it better. And so the end result of all of this is what we're trying to do is when the harvest area is in the open status, the water quality is suitable for the harvest of shellfish. And that means that any shellfish that you find at the store or restaurant, are guaranteed to be harvested from an open area. And I went a little quicker than I thought, but I am now open for questions if anybody has any.
We have one that came in from Carolyn Johnston. If oysters have been harvested in the 24-hour pre-warning time, they're theoretically contaminated. So are the shellfish discarded?
So if we close at sunset, but people have been harvesting that day, is that the question? I think so. The program is designed to kind of take into account for that. So that's why once we see the exceedance, we have till sunset. We have to give guys, if they're already out there, we have to give them time to finish harvesting. And we also have to give FWC a heads up. And so we don't do like the second we see an exceedance, we close it at 10:30 in the morning. And the program's pretty conservative to kind of buffer that out, that on a day exceedance that because the program is pretty conservative, I mean, we haven't really had issues in the entirety of this program for that. So that's kind of just how it's set up.
I also see a question from Jonathan Goodson asking how you monitor or measure pH.
Yeah, so we use a YSI and then we do our QA QC beforehand before we go out. So we do all of our SOP quality checks before we go out in the field, and then we do them when we come back to make sure the pH is, measuring properly.
And what can you tell by pH? What sort of an indicator is it?
For pH, so we don't really use salinity, temperature, dissolved oxygen, or pH. I mean, those are kind of gold standard parameters to get when you're out taking water quality sampling. If we are, sometimes we'll try to use them to mostly temperature and salinity. If we do start seeing illnesses, we do get some Vibrio cases here and there. That's just, a natural bacteria out there. This program isn't, this classification program isn't really designed to account for Vibrio, but we'll look at those sort of parameters to see if there's a trend. Like, is it, is the water temperature really warm? Is that why we're seeing this? Because sometimes we'll get them in months that we're not expecting to get illnesses. So we use it more on the illness investigation side than we do on how we classify our harvest area. Like, our program is really fecal coliform driven.
Trying to see some of the questions. Hang on, I'm having trouble seeing them.
We had another question from Gail Johnson come in that says, can we get access to water test history for their lease area?
You can. So we worked very hard, and I think we finally got a really good routine in. We have all of our water quality, actually probably you should add this to this presentation, on our website. Before, we didn't have it available until last year. And so we worked, there's a project going out of South Carolina where they wanted to combine the Southeast District Shellfish Program water quality data. And so they took it, they did a huge QA/QC on it. We came back, we did even more QA/QC on it. It is on our webpage under area information. Our website isn't as easy to navigate the easiest way would be to just Google Division of Aquaculture, Florida, go to Shellfish Harvest Area Classification Program page, area information, and then there's CSV files for every harvest area in the state. Each station has a GPS coordinate with it. It has all of the surface and bottom parameters. It has a fecal coliform. And then you can, we also have an interactive map where you can see the station that's closest to your lease. And then you can kind of filter. You have to be a little bit of knowing databases. It's the CSV, but it's, you can open an Excel and then you just sort it by station, find the stations closest to your lease or around your lease, and then you can see what the water quality looks like. And that goes all the way back to like 1980 or so, depending on the harvest area. And they'll be updated every 6 months. So I kind of gave me a goal of by the end of January, so you have all of 2021 data up there right now, and then the end of July. So we're going to update it every 6 months. We just thought that's when we do our QA QC. I thought that was a good time to update the online data.
Kennedy, do you see Gail's question?
Gail's question was the one I just asked.
Oh, sorry. Then we have Carolyn Johnson and
we asked Carolyn Johnson's also, but I wanted to ask if hers was addressed. I've unmuted you, Carolyn, if you wanted to chime in, if you got a sufficient answer or not. You may have to unmute yourself. All right, well, I assume that means it was answered well.
I've tried. Can you hear me now?
Oh yes, there you are.
Okay. Yes, it was answered. And then I had another question. If
Okay.
there's a repeat source of fecal coliforms in a particular area, how is this addressed and corrected? Do you have any power to do that, or is that the municipalities?
Yeah, I haven't really, so I've been here for 15 years and we haven't really had like a specific problem station. I know in the past, like in some areas that were heavily septic tank, we'll be advised to try to do a dye study, but you have to go to the property owner and say, hey, can we do a dye study? We think your septic tank is faulty and now it's causing water quality issues. I haven't had to do that. We are having issues in one of our bay systems in Bay County. It's kind of the lower part of East Bay. And that's exactly where Hurricane Michael went through. And so it took out all the trees. And so the lower section of that area is actually getting worse. And we have it closed right now. We'll reassess it. And so sometimes we just close the area. Sometimes we'll reclassify it and we'll downgrade the management plan. But I would think if we knew the direct source of the problem, it's usually septic tanks, faulty septic tanks. We have those buffer zones around wastewater treatment plants and marinas. We might try to work with the municipality and see if we can work with the individual owner. And a dye study is usually required to, kind of prove that that's the problem. But after that, I don't know if you can really have to do anything about it. Sometimes it just leads us into reclassifying our harvest area.
While they continue to put the pollutant in.
Right.
Gotcha.
I don't have direct authority, to kind of make them clean up. I think we would probably go to DEP and FWC. And if Department of Health is more appropriate, then we might work with them too and see if there's.
Gotcha. So it's more up to the municipality to enforce.
Right. But that's a good question. I may have to actually like look into that a little deeper to see, you know, if it's a municipality issue. I'm sure a city or town would probably want to work with us. I just don't know how it would work if we got down to an individual level.
Thank you.
I'm kind of happy I've never had to deal with that. That'd be a hard one to navigate.
Yeah, Apalachicola Bay is pretty lucky. We don't have a lot of point source, which is like a factory or farm nearby. A lot of ours
Right.
is done conditionally because it's a lot falls into the floodplain and comes down. And people, when they think of fecal coliform, they think immediately of sewer and sewer plants malfunctioning, but we don't have a lot of sewer plants directly related to the river anywhere nearby. So
The wastewater treatment plant's land-based, and so we don't really have a wastewater treatment plant surface discharge in that area. So that's positive.
Yes.
I think the main input of fecal coliform, it takes into account wildlife too, is the river.
Yeah.
That's what drives that system, and wind and tides. It's a really hard area to classify because of the north. Like we're actually getting ready to start the survey for Appalach. And I haven't decided if I want to keep it the same and relook at it in 2 years when, you know, we have a better idea what the wild resource is looking like. Because we're gonna have to manage kind of, you know, all the activities, which makes it also kind of hard, and you have to get really strategic about it. Because you don't want to impact the leases, but you don't want to impact the wild harvesting either. You want to be this synergy between the two. And so there is a really big north-south, east-west influence. You can kind of see it. You know, I think maybe this, like a learning activity for me, if I wanted to be really specific to Apalachicola, I could put up what the management plans are for each, and you could see how they get better as you move away from the river. The ideal spot to put leases in improved areas. So.
Jonathan brings up a question about marinas, and I know this is the reason why many marinas have been denied in this area because of the wild harvest. But Jonathan asked, wondering about liveaboards in marinas. Many of them never move. I would guess they are pumping overboard at night. Is this something you address or FWC?
So for marinas, what we have done, most of our marina buffer zones are based off of the total number of slips in the marina, whether they're occupied or not. And thankfully in this area, from what I know, is there's not a whole lot of liveaboards. I think I read an article, I don't know enough about it, about the marina down in Appalachia. They're not going to allow liveaboards anymore. But if there's a problem, they should have a pump out sewage system there that people can pump out. We look at all of that. Most of the major marinas that allow liveaboards have a pump out facility that boats can discharge their waste into. So we look into all of that. But since the buffer zone is based off of the total number of slips, and say you had 4 liveaboards in there and they were illegally dumping their waste, our buffer zone is so big because it's not based just off of those 4, it's based off of every slip. And so our buffer zones are pretty big. And we do worst case scenario. So we fecal coliform at a raw stage is 2.9 times 10 to the 7 colony forming unit. So it's pretty big. So we use the maximum amount of concentration of what you would see with a raw discharge. And so our buffer zones take into account, if there was an accidental discharge, it would be diluted out before it got to any area that was allowed harvesting. That answers the question.
Yeah, you were correct. You did read an article. Apalachicola did is now not allowing liveaboards at Battery Park Marina. So
Right.
we really don't allow liveaboards at any marina at this point. They haven't been allowed, but there have been some. And there is a pump out at all of them. And it is hooked to the sewer system, which is a good thing. So.
Right. Because then it's not contained on site, like a septic tank, which then could also fail. Yeah, like we classify pretty conservatively with wastewater treatment plant, surface water discharges, and marinas. That's just how we did it historically. And it makes sense, you know, especially in a sensitive area like Apalachicola too. So.
Okay, well, if there are no more questions,
We actually just had one pop in from Chad Hansen.
go for it.
Does the abundance of oysters in the system have an impact on the duration an area is closed following a rain event? Considering the depleted level of oysters in Appalachia, for example, does this have an effect on the filtering capacity to reduce the bacteria levels?
So for fecal coliform, I don't think we've directly linked it to oyster populations declining. The retention time of fecal coliform in the water column or at the surface is really a lot dependent on the amount of daylight because of the UV. UV kind of kills it. And so that's why we kind of see better water quality in the summer. These are the trends I've seen sort of looking at data sets. And this is pretty much true in every harvest area. We kind of see the water quality degrade in the summer, in the winter, not to the point where we have to close things down. And then it kind of gets you know, it gets better as we go into the spring and summer. So when we build these management plans, we look at a 12-month period. And so it kind of averages, the management plan kind of averages between winter water quality and summer water quality. And so if we only looked at summer data, we could probably have management plans where they would never close unless we had, you know, 7 inches of rain. In the winter time, they would close on, you know, half an inch of rain. And so it kind of takes into account those different seasonal differences you can see. Some areas do have summer or what we call spring fall management plans, and then we'll switch over to a winter management plan where we'll close on less amount of rain. We only have about 5 areas that have that. It kind of makes the managing of the area really complicated. In areas that have leases are really, you know, a lot of wild harvest. The harvesters don't really want a seasonal management plan like that. So we take into account a lot of different things like the activities that are going on and sort of these monthly seasonal trends that we see within fecal coliform. I don't think, I haven't seen any studies that directly link, you know, oyster population decline to increased fecal coliform levels. But it could be impacting other things like algae and harmful algal blooms. There's been talk about, you know, shellfish restoration on helping, especially in Southwest Florida, for the ongoing red tide blooms that seem to be happening on an annual basis.
Yeah, we hope those stop.
Well,
I have a feeling
it's
going to increase with global warming. So
I know we just went through, I would think, probably one of the worst blooms I've seen as it impacts harvest areas. Maybe not the duration, you know, from 2020 to 2021, but at one point we had harvest areas from Bay County all the way down to Tampa Bay closed pretty much. And so a lot of leaseholders, a lot of, you know, wild harvest areas were, most of our wild harvest is coming out of the Big Bend right now. But it impacted all of that. And so that didn't make for a very fun fall last year, but we got everybody open pretty much by Christmas. So it is pretty, I heard St. George Island got it, you know, some respiration for a while. And, you know, that's never anything good to be around. It's pretty miserable. I've been in a red tide bloom taking samples and it's not fun.
yeah.
So,
Okay, if we don't have any more questions, I'd like to say thank you, Jill, so much. And Kennedy,
welcome.
Kennedy Hansen has been our tech on this call.
Thank
And we
you.
appreciate you coming, all of you attending the symposium. It has been recorded and will be available on the Friends website at apalachicolareserve.com. We won't have a Sci Cafe in February because we're having our annual symposium, which if you're like me, it's over my head, but really enjoyable presentations on February 17th. And we will record those also, but a lot of really exciting science going on in the Bay. We will return in March 24th. We'll have a Sci Cafe with an expert and a botany expert who now lives on St. George Island, Jim Folsom. So be sure to join us for that. And we appreciate you coming.
Thank you for having me.
Okay. Thank you.
I'm not sure if I should click out or if you